Okay, Section 3, Chapter 2, Buffering Against pH Changes in Biological Systems. So we know in a titration curve, there's going to be a region within that, there is a buffering where it levels out before it takes off again, that lag. So in chemistry, you've utilized buffers. Usually they're labeled, buffers, pH 10, buffer pH 7, buffer pH 2. A lot of times we forget what that's actually happening. Well, within that buffer, whatever it is going to be constant. It will be a compound that at given pHs will give up or accept hydrogens, okay, depending upon whether they're weak or strong acids. Most buffering systems are going to go with weak acids because you don't want them to give up, but when they hit that certain point in the pH, you want them to accept back. So you want to try to find the buffer that fits within that region. Remember where the titration curve goes and comes, that lag before it takes off again. So whatever you're trying to keep within that region, there's optimal. That's where you want to be. Now, are the buffers going to sit there and maintain a buffer of 4.7? No. There's going to be fluctuations. It's going to go up a little, but then all the hydrogens are going to be released again because it's going back up, and then it's going to drop, and then they're going to be taken back up, and so you're going to set this balance. So it's the average balance within that buffer region. Well, what you see there happening in that titration curve within that buffering region is what's shown here. Acetic acid, acetate. They are in balance within that region. pH goes down, goes to acetic acid, pH goes up, goes to acetate. So you're looking at this Teeter-Totter effect. What's happening? See here? When the pH is changing, you're going from acetic acid to acetate, hydrogen ions released. A hydroxide ion nearby will take it up, form water. But things get buffered back, so all the free hydrogens are taken up, pH goes, boom, starts to go back and forth, back and forth. So you go more acidic, acetic acid to acetate. I said that backwards a second ago. pH going down, hydrogen given up, taken up, pH goes back and back and back and back and back, as you're basically just cycling through, as long as you're in that buffering region where things are trying to stay balanced. Now, if you actually want to sit there and discuss these rates of what's occurring and when and how, that's what this Henderson-Hasselbach equation is all about. pH equal to pKa. Remember, as I said, they're kind of plus the log of acid and the beginning concentration of whatever. So that would be acetate over carbohydrate. You can go through and you can prove this. Ka is the concentration of Ha over Ha. You go through, do the algebra, move things back and forth, multiply both sides by concentration of hydrogen, or I'm sorry, the inverse of acid over Ha. Take the negative log of both sides. Well, negative log of concentration of hydrogen, that's pH. Negative log of Ka, that's pKa. Minus concentration of that. So when I sit there and say the pH and pKa are roughly the same, Kind of, sort of. Concentration is going to be a little off. So what? What's this have to do with, cost of gas in the morning? Well, not much, really. Until you take into account that, pKa, pH, kind of, sort of the same thing in, what's happening. Concentration of free hydrogen ions in a solution. And as we discussed last section, we want to maintain in our cells and our bodies rough pH. Not too high, not too low. Things go bad. Acidosis, alkalosis, and so on. But that's okay. Don't have to worry about it, because we as living things have spent the past 2 billion years evolving to have inherent buffer systems. To help maintain the pHs of interstitial fluid, blood, cytoplasm, within our vacuoles, within our organelles. Great example you see here with histidine. Histidine, pH 5 versus pH 7. As the free hydrogen concentration increases and the pH lowers, histidine, the nitrogen in the ring there, will associate with the hydrogen. Take it up. It's now associated, it's not free, thus pH now goes back up towards 7. If it goes to get acidic again, boom, back up. Now, is this perfect? No. But this, along with some of the other mechanisms we're talking about, are how our bodies, our cells, our being us living things, prokaryotes, eukaryotes, you, your dog, your cat, the plant outside, whatever. We use that, things like the histidine and the amino acids. We also use phosphate buffers. You take dihydrogen phosphate, dissociates, you get free ion, free hydrogen ion, and hydrogen phosphate, monohydrogen phosphate, depending on who you're reading. H2PO4, proton donor, HPO4, proton acceptor. They just keep going back and forth, back and forth. You're going to find this in the cytoplasm of most living cells. Free hydrogens are not a good thing to have. Free hydrogens have a tendency, as I said in the last lecture, to interfere with many, many things. Anything held together by hydrogen bonds, now I have something that's going to interfere with it. Loss of protein structure. Start stripping away at, interfering with DNA structure, mRNA structure, our ribosomal RNA structure, things like that. Also have bicarbonate buffer system. You can see this in the blood. You're going to have bicarbonate flowing in our bloodstream, taking up, releasing hydrogens as needed to help maintain an almost 7.4 pH. One's the donor, the other's the acceptor, depending upon where the pH is going. Too high, give it up, bring it back down. Too low, take it up, and back and forth. So when you look at this, bicarbonate and carbonic acid, back and forth, back and forth. A lot of this is going to be driven by CO2 levels in the blood. CO2 with water in the blood gives us H2CO3. One of the things we seem to forget is, yes, you breathe in oxygen, but you're also breathing in carbon dioxide and other things. You're exhaling some of the carbon dioxide and you're retaining some of the carbon dioxide. That CO2 that's dissolved in your blood, yes, you're going to get rid of it via your lungs, some of it. Some of it's going to stay. Some of it, as we see here, is going to be used to help buffer blood. How much is going to stay is going to depend upon the partial pressures of CO2 in the atmosphere you are breathing. How much is out there? How much is passing back and forth, in and out through your lungs? Bicarbonate buffer system is going to be there to help maintain a pH, as I said, of roughly 7.4. Yes, it will fluctuate up and down depending upon your breathing. Are you breathing heavily? Are you not breathing at all? Are you getting enough gas exchange in your lungs? What's the pressure? Are you at 1 atmosphere pressure? Are you up on top of a mountain? Are you down, Death Valley, well below sea level? Those will have an issue. Those will impact the issue. So as you see here, it's not all about just CO2 being there. It's how much you're taking in, how much you're giving out. You're constantly making CO2, so yeah, you're going to have enough. But if there's not enough in your environment, or there's too much, too little, it's a Goldilocks thing. Too much outside, it's going to be going into your lungs. You're not going to be getting any out. It's going to mess with maintaining pH. You're up on top of the mountain, so there's too little. You're going to be losing a lot, which means you're going to have issues with maintaining the correct pH level. Ask anybody who's climbed top of mountains, and I'm not talking little hills like we have around here. I'm talking top of the Rockies, Himalayas, oh crap, can't think of the European mountain chain right now, but I'll probably remember it after I'm done lecturing. Get up top there, high up, high altitude, where atmospheric pressure is well low, our normal 1 atmosphere at ocean level. CO2 doesn't always go in, all of it comes out. Well, you're going to have trouble. You're going to start running more alkaline or acidic, depending upon you're too high, you're too low, where you're at. And again, there is a constant hydration of CO2. How much is there? How much is necessary? How much, what's the keeping things in balanced rate? And no, we're not going to go through that. As you see here, somebody went through and did an example of all the algebra, the math. So when you go and you start looking at, pK, pK combined, K combined, things like that, it allows you a backdoor way of calculating what's required at different atmospheric pressures, CO2 level, to form the bicarbonate, things like that, to maintain a specific pH. If you can sit there and calculate how much is necessary CO2 levels, you can then go through and then back calculate what the pH has to be, approximately 7.4. And that's how we got the number. If I go and I bleed you right now and sit there and try to use a pH paper, it's going to be around 7.5-ish, in that range. But you also have to take into account immediately as soon as that blood hits the atmosphere, things change. CO2 concentrations change. Gaseous oxygen changes. Proteins change, deproteinate, and things like that. So when you see 7.4 listed in textbooks, a lot of times this is how they came to it. They back calculated it. And it turns out everything in your body operates at specific pHs, optimal pH. Yeah, 7.4 is your bloodstream. That's all the blood proteins work best. But not everything in your body works best at 7.4. You look here at this graph. Pepsin and trypsin, 2 digestive enzymes. We use these to break down proteins. Pepsin is going to be found in your stomach, trypsin, your small intestine. Turns out stomach is a pH that's 3-4 logs lower than small intestine. Small intestine is weakly acidic because you're wanting to absorb, not digest away most of the things by the time it hits your small intestine. So pepsin, as you see here, the black line, its optimal activity, the peak, is going to be down 2 and below, which is about where the stomach is. As you go up the pH scale, becoming more basic, pepsin starts to unravel. Pepsin basically loses its three-dimensional shape, loses the function. It denatures. Now as you get up closer to 4, and you start to see this is where trypsin starts to take off, starts to work better. Optimally works between pH between 6 and 7. Guess what the pH of your small intestine is? Between 6 and 7, depending upon what region you're at. Trypsin, if you were to go and drink down some of it, well, look at it. It's not going to operate very well in your stomach, and actually it's probably going to be broken down by pepsin. Pepsin, since it's denaturing because of the higher pH of the small intestine, it's going to be broken down by trypsin. Everything works at a specific optimal pH for that molecule, for that macromolecule. What happens though when all this stuff goes awry? Well, we see that nowadays in diabetes mellitus. Okay, diabetes mellitus is one of the offshoots of diabetes that we see, issue that many diabetics will run into. They have trouble maintaining that pH of roughly around 7.4. Too high, too low. Enzymes can't function. Enzymes start to break down. Cells and tissues start to break down. Diabetes mellitus, more often than not, these patients are going to have an issue where their blood starts to get down to 7.3. Maybe a little lower depending on who they are, what's going on. They accumulate beta-hydroxybutyric acid and acetoacetic acid. Now, under normal conditions, beta-hydroxybutyric acid and acetoacetic acid are going to be further broken down, or they're going to be protonated so that they are not, lowering the pH. But in diabetes mellitus patients, that's not occurring. So they accumulate, and you start to see the lowering of the pH, and it's not much. I know a lot of people sit there and go, but it's, 7.4 is where it should be, and it's only 7.35. That shouldn't be too bad. But it's enough. We're not talking battery acid in their bloodstream. We're just talking just enough of a change. We're out of that buffering that we talked about, that buffering region, so that proteins denature, lipids, which are making up your membranes, get denatured, don't function correctly. Things just go off.